In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory. My name is Som Shuvra, and my role as a technical information scientist is to serve the research community with in vivo research education solutions and offer answers to all your technical queries. For anyone watching live, please use the comment section of this video to share with us where you're viewing from and any questions that you have. We'll try to answer them live today. So today we have our guest, Prabir Banerjee, on the show. Prabir and I together look to discuss the Jackson Laboratory's graft versus host disease platform and how it might help accelerate your research goals. First of all, welcome to JTT, Prabir. It's great to have you here, and could you please introduce yourself to our audience?
Thank you, Som. Hello, everyone. Hello, viewers. Good morning, good afternoon, wherever you are. My name is Prabir Banerjee. Joined JAX 8 months back as the product manager for in vivo services. I come with a background of an MBA in life sciences and 8 years of experience in product management, which is across the pharmaceutical and cell and gene therapy space. My role here at JAX is to understand the in vivo services, the products that we have, and then also to understand that how can this be brought closer to our customers, and how can we understand them better, their needs, and modify our products and bring them back to the market. So that's overall the gist of my role. And thank you for having me over here.
Thank you very much, Prabir. It's great to have you again on the show today. So for all of you who are new today, we did have a JAX Tech Talk on the very similar topic last year, where we discussed various factors associated with assessing the efficacy of GvHD therapeutics. The talk was titled, Let's Talk, Assessing Efficacy of GvHD Therapeutics. This was aired back in September of 2021. I would highly encourage all of you to revisit that talk when possible. Now, as a follow-up to that great discussion from last year, let's start this discussion today, Prabir, with my first question to you. Where do you see the JAX's GvHD platform being used in the drug discovery pipeline?
Yes, so before even I speak about the application of this platform in the drug discovery pipeline, I would just like to brush up on what GvHD is. So basically, GvHD stands for graft versus host disease. It's one of the major complications that you would see following an allogenic hematopoietic stem cell transplantation, which are usually done for treating blood cancers and various other genetic disorders. And what happens is it's the donor cells who start attacking the recipient tissues and cells because they consider them as foreign. And that's where graft versus host disease comes up. So coming to your question, where is it applicable in the drug discovery pipeline? So let me bring up the slide that would explain this better. So can you see my slide?
Yes, I can.
Okay. So basically, the GvHD platform belongs to two big buckets that Jackson Laboratory is an expert and leader in. I would call them as the Humanized mice model and immunology segment. So the GvHD platform uses PBMC-based human donors as the base for this platform, and it's also called as a xenograft model, and can be applied in the target identification, target validation, and lead selection and optimization stages, as you can see on the screen. So it's the initial stages of the drug discovery pipeline where this platform can be applied. So any customer or client who is looking to kind of screen their efficacy of their novel immunotherapeutic drugs or immunomodulatory drugs that they want to use to treat cancer or autoimmune disorders, that's where they can look to come to us and inquire about the GvHD platform, where this would be the perfect platform for them to test this efficacy of their candidates.
Excellent information, Prabir. I'd also like to supplement what you mentioned in terms of utilizing this JAX GvHD platform. When you sign up for a discussion with us, you can get project support from various study directors The assistance can start from the moment you reach out to our team with a small preliminary idea, perhaps, and that can get molded even or translated into a much bigger experimental thought. The discussion can encompass from initial study design to full-fledged report analysis. The JAX team has extensive experience conducting large-scale GvHD studies like these for various types of clients. And having said that, Prabir, can you share with our audience about the advantage of using the JAX GvHD platform in terms of translatability to human GvHD, perhaps?
Yeah, and that's one of the big advantages that the GvHD platform provides to our customers, is that it's a very clinically relevant model, very much validated, where we can actually show or model the human GvHD into our mice model. So let me go into the next slide, and I can give some background to it before I even explain the slide. So what we try to do is we use the NsG mice, which is like 6 to 8 weeks old, irradiated and then engrafted with the PBMC donors. So as soon as they're engrafted, the cytokines get activated along with the antigen presenting cells, and this is followed by T cell subset expansion. So and after that, what happens is what you see on this slide. So this is an internal study that we did on an humanized PBMC NsG mice, where you would see that the initial days, that is at day 5, you would see greater percentage of CD4 cells. But as we move ahead into day 18, you would see more CD8 cells. So that's when we say that the CD8 T cells are trafficking towards the tissues, which is ultimately causing damage to the tissues. And this is the sign of GvHD that we are talking about. And this is exactly what happens in a human when they are undergoing any sort of transplantation.
I agree, Prabir, and this is very helpful. And I want to add something to what you just mentioned. Other than what, Prabir, you're showing, very importantly, all our researchers can also stand on the shoulders of the industry-leading NsG mouse portfolio that has been well studied and well published on. NsG mouse model variants are among the most highly immunodeficient mice and the models of choice for various xenograft modeling, humanization experiments, and other types of disease research. The compound immunodeficiencies present in these NsG mice actually permit the sophisticated modeling of many areas of disease biology and thus help to support the efficacy testing of various cancer therapeutics. So basically, in short, the advantage of JAX's knowledge of the mouse model itself can also be very helpful in this context. We have many resources and published literature to share that with you that can cite similar experimental setups like that you have in your mind. Having said that, another very important question we get very often, Prabir, is how reproducible is the JAX GvHD platform?
Yeah, I totally understand the importance of that question. So I would like to answer it again through one of our internal studies that we have done that you see on this slide. So reproducibility of PBMC donors across studies. So on the left-hand side, what we have done is we have plotted the survival curves of 4 PBMC donors. And these survival curves are basically an indication of how long a humanized PBMC mice has survived once they were injected with PBMC donors. So as you can see, all 4 of the PBMC donors have a very unique profile in terms of their survivability pattern. So it's a very donor-specific platform, which is reproducible, that we offer as an advantage to our customers. And speaking of reproducibility, to what we have done on the right-hand side, if you see, we have taken one of the donors, which is 213, that we have shown on the left-hand side on the right-hand side graph, where we have used it in 2 separate studies. And these studies were done independently at 2 different times. And again, if you see, the survival pattern of donor 213 is very similar. So this is the kind of reproducibility that we are talking about, that JAX's GvHD platform brings in. So any client who's coming into our organization and wants to perform a GvHD platform, this is what we can offer them. And since we are talking about survival curves, I think it's also important to highlight the next advantage that this platform provides is is that the PBMC donors that we have, or JAX has, is all pre-tested and pre-characterized. We have a large inventory of PBMC donors. And when we say it's pre-tested and pre-characterized, that itself is a big advantage to our prospective customers or current customers, because they don't have to do this in-house. So that there itself, we are saving a lot of time. And then since it's pre-tested, so we know how the survival curves would be, and it's pre-characterized, so we know what's the GvHD progression like. So this is the kind of advantage that the client would get right at the beginning when they come into JAX. They can pre-select the PBMC donors based on the study that they want to do. So this eventually would save time and help us determine the timeline of the study, of how long a GvHD study would run. So these are the two big advantages that Jackson's GvHD platform provides to our customers.
Yeah, very, very, very powerful, right? Because customers can save a lot of time because of this already established knowledge that we have. So having said that, Prabir, the next question that we get a lot from researchers is looking into additional components into the study design. Is that something that can be done? Let's say to understand the association of GvHD and cytokine release syndrome. Is this something that can be done on the JAX GvHD platform? So essentially, can CRS be incorporated into a study like this?
Yeah, that's an excellent dimension that you have brought into this discussion, because we have had a couple of experiences where such discussions have come up with customers. And I can explain this better by bringing up the next slide, which is similar to the first slide that we just spoke during this talk, is that how GvHD platform can be used from target selection, target validation to up to lead optimization. So that's the stage where you're screening the efficacy of your drug. But the next step is you want to test the safety and toxicity of their drug. So what you do is you can always come up to Jackson Laboratory and use the CRS platform that we have recently launched. It's again an innovative platform, again based on PBMC donors. So as I'm saying, the base of both these models are same. It's based on PBMCs. And once a client comes in, what they get from us is efficacy, toxicity, and safety data all at once, right from the start till the time they have to go ahead and file an IND. So it's very easily can be said that we are providing a full spectrum of platform over here, where we are providing all the 3 data points at one go and saving time for our customers to go ahead and file an IND. So that's the kind of complementary package that we have in JAX, where GvHD complements the CRS platform.
I must say this is really, really useful. Let me look into the live questions now, because we see a few questions come up. So let's see. So there's the first question here, Prabir. What is the typical GvHD study duration in this context?
Yeah, that's a good question. So basically, I spoke about pre-testing and pre-characterization of PBMC donors, which eventually helps in ease selection of PBMC donors. So this particular step basically helps us a lot in saving time for our customers as well as for ourselves. And this eventually helps us to complete any study. I would say on an average, the time to completion is around 35 days, which is one of the best in the industry. So that's the kind of timeline that we can always commit to our customers who are coming to us for their GvHD needs.
Fantastic. Let me see if there's another question here. So what positive controls could a JAX GvHD efficacy study include? I think this is a very important question, and maybe I can take this first and you can add to that, Prabir, later. So yes, you can certainly consider including a control arm using a clinically approved immunosuppressive drug. Let's say, for example, something like abatacept. Abatacept, which is a known immunomodulator that decreases the T cell expansion and improves survival in a GvHD model. So let's say if you're looking to investigate a novel immunotherapeutic by utilizing our JAX GvHD platform, we can test the same in this platform and have that efficacy compared to abatacept to understand the mode of action with readouts such as midpoint and terminal blood sample collection, survival curves, and in the end, of course, as I mentioned earlier, a detailed study report. Do you want to add to that, Prabir?
Yeah, I would just add that we have done several studies using abatacept, and we have that experience in-house within our team. So I think you covered the rest pretty well on that. So I would just recommend whosoever amongst our viewers are interested to know more about it, they can feel free to reach out to us to learn more about the data sets that we have in-house around this positive control.
I think that's all the time we have for questions today. So thank you again, Prabir. This conversation was really informative and perhaps helpful for all of you to have a refurbished understanding of the JAX GvHD platform and how it can supplement your research needs. We hope that some of the things we discussed today would stimulate additional questions, which we would love to hear from you in the long run and keep the discussion going forward. That's all of the time we have again for this week. Thank you for joining all of you for JTT number 51 this week. Our next JAX Tech Talk is scheduled for March 15th, 2022. Please follow JAX Tech Talk on LinkedIn so you're notified anytime we have a new episode or we share a new content, and also subscribe to our playlist on YouTube. This is Prabir and Som signing off and wishing all of you stay healthy, stay safe, and stay excited about research.
Thank you.